Good evening, everyone. So today we will present on canine hepatotoxicity. So my name is Dushan Ramachan, matric number 181290.
My name is Dongwei batch number 181588.
My name is Siti Suhaila with your Awaro, metric number 181636.
My name is Putri Sabina with the metric number of 182201.
My name is Ek Indian with the metric number of 183825.
My name is Amir Abdurrahman, metric number 182082.
Yes, so we were given this topic of discussion and we've got to discuss it in the flow. So we came with some conclusions. So we first found out that canine hepatotoxicity is a lysosomal storage disease affecting dogs. This type of storage disease of inherited metabolic disorder that results in malfunctioning of the lysosome. Now, what is lysosome? Lysosomes are membrane-bound organelles, spherical vesicles containing hydrolytic enzymes. These hydrolytic enzymes are capable of breaking down all types of biomolecules, including proteins, nucleic acids, carbohydrates, lipids, and cellular debris, and pass the fragments to other parts for recycling. So basically, lysosomes are like the recycling center of the cell. So the copper accumulates, prevents the lysosomes from functioning. They can't recycle it, and hence it accumulates. They can't remove the copper accumulation occurs, malfunctioning of cells, and disease. So when one of the enzymes is defective because of the mutation, the large molecules accumulate within the cell, eventually killing it. For our case, we concluded that it was canine hepatotoxicity is the accumulation of copper in the liver of the dog that leads to progressive damage and scarring of the liver. Disease mostly affects Bennington breads. We couldn't figure out on our discussion, we couldn't figure out the enzyme that is deficient and why only Bennington is physically Bennington terrors seems to be affected by this. So then we've got to search for it, and my friends search on different types of different aspects of the disease to find out more about it. So some of the, you want me to tell the discovery?
No.
Okay, anyone proceed now with a general explanation of the lysosomal system?
So, as Sasha has mentioned just now, lysosome is a membrane-bound cell organelle containing hydrolytic enzyme known as lysozyme. And they are known to contain more than 50 different enzymes, which are all optimally active in an acidic environment of about pH of 4.5. So thus, lysosomes act as a waste disposal system of the cell by digesting unwanted materials in the cytoplasm, both from the outside of the cell and also absolute component inside the cell. For this function, they are popularly referred as the suicide bag or the suicide sac of the cell. Furthermore, lysosomes are responsible for cellular hemostasis, for their involvement in secretion, plasma membrane repair, cell signaling, and energy metabolism, which are related to health and diseases. Depending on their functional activity, their sizes can be very different. The biggest one can be more than 10 times bigger than the smallest one. So for the formation of lysosomes, enzymes of the lysosome are synthesized in the rough endoplasmic reticulum. The enzyme released from Golgi apparatus in small vesicles, which ultimately fills with the acidic vesicles called as endosome, thus becoming the full lysosome. In this process, the enzymes are specifically tagged with the molecule mannose-6-phosphate to differentiate them from other enzymes. So lysosomes are interlinked with three intracellular processes, namely phagocytosis, endocytosis, and autophagy. So extracellular material such as microorganisms taken out by phagocytosis, macromolecules from endocytosis, and unwanted cell organelles are filled with lysosomes, in which they are broken down into the basic molecules to be digested. So thus lysosomes are the recycling unit of the cell. For lysosomal storage disease, synthesis of lysosomal enzyme is controlled by the nuclear gene. Mutation in the genes for this enzyme will lead to genetic diseases, which are collectively known as the lysosomal storage disease. These diseases are due to deficiency in single lysosomal enzyme that prevents breakdown of target molecules. Consequently, the undegraded materials accumulate within the lysosome and target molecules, and often giving rise to severe clinical symptoms. It causes organs to function less efficiently, resulting in progressive deterioration in physical and mental state and eventually death. So the symptoms of lysosomal storage disease vary depending on what type of cell it is malfunctioning and what material is being stored. Some have a relatively early age of onset, others may not appear until middle or old age, and symptoms range from fairly mild to severe and deadly. Storage disease symptoms can include developmental delay, movement disorder, seizures, dementia, deafness, and blindness.
Okay, so now, so as we know, canine hepatotoxicity is definition of copper in the liver.
Yeah.
So before explaining about the disease, can you want to explain about the normal liver function?
Yes, I will explain about how can liver functioning. Well, actually, the canine liver function is actually the same as almost the same as human liver function. So we will be familiar with it. So anyway, I will explain it. So a dog liver, a very complex and hardworking structure. The liver will filter the blood and has hundreds of other functions, most of which are necessary to life and are not done elsewhere in the body. Every part of the liver is capable of performing all of its tasks. The liver has an amazing ability to recover from injury and regenerate new tissue. And all the blood that will leave the stomach and intestine will pass through the liver. The liver processes this blood and breaks down the nutrients, chemicals, and drugs into forms that are easier for the rest of the body to use. There are more than 500 vital functions associated with the liver. Some of these functions include the production of bile, which is released into the gastrointestinal tract to help break down fat in the small intestine during digestion. The liver makes bile continuously even when food is not being digested, and extra bile is stored in the gallbladder. Liver also responsible for the production of certain proteins that circulate in the blood, such as albumin and several factors responsible for blood protein. It also produces cholesterol and special proteins, such as lipoproteins, that help carry fat through the body. The conversion of excess glucose sugar into glycogen and the storage of this glycogen all happens in the liver. Glycogen can later be converted back to glucose for energy whenever it is needed, which we had learned in biochem class. So the storage of blood that can be shunted immediately into the general circulation when needed following injury or sudden blood loss. The regulation of blood levels of amino acids, which form the building blocks of protein, is also made by the liver. The storage of iron that is used in the processing of hemoglobin in the blood, and hemoglobin is essential for carrying oxygen in the blood. Other than that, we also have the conversion of ammonia in the blood to urea, like we discussed in the last video. The bacteria of the intestinal tract produce ammonia as they break down proteins, and ammonia can accumulate in the blood at potentially toxic levels. Urea is a second product of ammonia and is excreted in the urine. Clearing of the blood of foreign substances such as medication and anesthetic agents that are administered to animals also happen in the liver. Resisting infections by producing immune factors and filtering bacteria from the vaccine also happen in the liver.
So thank you. And then we're going to proceed with the disease liver, the explanation of canine hepatotoxicity. So anyone can give?
Okay, copper hepatotoxicosis in dog is a condition caused by an abnormal accumulation of copper in the animal liver. To be in this topic is the canine, resulting from either inflammatory hepatic disease or an inherited metabolic defect, which can lead to progressive damage and scarring of the liver, which is called cirrhosis. This condition may be secondary to a primary disease, which the effect from other diseases, or it is purely from inherited genetic-based diseases. So, causing hepatotoxicity from secondary diseases is so rare, and it is more clear to be found in genetic inherited, such as the Bedlington Terriers, Doberman Pinschers, West Highland, White Terriers, Skye Terriers, and Labrador Retrievers are dog breeds known to be susceptible to this disease. This condition is more prevalent in females than in males. Findings in this breed share some similarities with the Bedlington Terrier disorder, but notable differences are evident. For example, progressive lifelong copper accumulation has not been demonstrated in the non-Bedlington Terriers breeds. So the cause, like I said just now, is because of the inherited gene. It is an autosomal recessive trait in Bedlington Terriers due to the lack of a specific gene with a nickname called COMD1, COMD1. It is coding for liver protein, which is the enzyme that is going to involve in the metabolic pathway of the copper metabolism. And it is the actual cause of this condition. It is the only cause. And at one time, possibly as many 2/3 of Bedlington Terriers either were carriers of the gene or were affected by the disease. So it's a real problem for these breeds. But with recent genetic screening, meaning like widening the gene pool or things, proper breeding, the incidence is now much lower. So I'm going to talk about a little bit about copper. Copper is a trace element and an essential micronutrient. Adequate amounts are required as cofactors for numerous enzymes, electron transport proteins, and antioxidant molecules, all of which are crucial for everyday cellular function. However, excessive ingestion, the derangement in storage, or compromised excretion of copper leads to copper accumulation. And in this case, which is the canine copper hepatotoxicity, it is due to the compromised excretion of copper because, like I mentioned before, the specific gene is COMD1 is responsible for encoding the enzyme called MER1, MERR1, and this is the last stage of the metabolic pathway of copper metabolism in the liver, and that this is where the part which it cannot MER1 acts like a bridge between the liver to the blood to be excreted, but this enzyme is not available, so it's trapped in the liver. Yeah. So such stress leads to free radical formation, lipid peroxidation, and DNA damage. So basically, it's toxic to the. So hepatocellular damage leads to acute, then chronic inflammation, and eventually to cirrhosis, and finally death. In breed-specific autosomal recessive inherited patients, the affected individuals are known to have inadequate biliary excretion of copper because of the gene deletion, which I mentioned just now. This gene encodes the small cytosol protein, as I said just now, the number one. The actual name is quite long, and if we're going to look to the process, it's really complicated. So since the overwhelming metabolism from biochemistry with this, I can't take it for the final process of vesicular copper movement. So and like I said, it's the excretion and the chemical membrane of the hepatocytes. To identify these diseases, affected Bedlington Terriers can now be identified by genetic tests, which is CT marker, CT deletion, and this is resource from Bed.
Before we proceed, okay, I'm not really exposed to dogs. I'm sorry. But how do Bedlingtons actually look like? How do they physically look like? Are they small or big? How do Bedlingtons actually look like? I'm not exposed to them.
Yeah.
They're actually medium size. So they're, how to say? Yeah, they're quite cute.
How actually big is it?
Yeah.
They like that.
I didn't see actually, but I searched it online.
Mhm.
It's okay. Sure.
Just medium size, like just like this. Yeah.
Okay.
So you can actually hold it.
Oh, yeah,
Ah.
actually, some fish are like really
Yeah.
big.
It's not like German Shepherd is very big. Just medium
Is it
size.
just like the UPH stuff?
Yeah, maybe yeah.
The standard. Okay, fine.
Standard size.
size of-
That's all right.
Okay.
So
Sorry.
yeah. So before we should continue, I would like to share a bit of information with you. So hepatotoxicity. Many of us get afraid by this long word. So just simply is the capacity of a drug or chemical or any other exposure to produce injury to the liver. So canine, dog, couple hepatotoxicity is the ability of couple to injure the liver, the ability of couple to produce injury to the liver.
Mm.
That's it. That's quite simple.
Yeah.
The word is quite complicated.
Yeah.
So then, Amir, thank you, Amir, for explaining about the disease. Then we should gonna continue with the symptoms. So let's see.
I'll explain about the symptoms of the hepatotoxicity. But before beginning, I will start with explaining about the classification of hepatotoxicity. The copper liver disease, medically referred as hepatopathies, is fall into two categories, which is primary and secondary. Where for the primary, it fall into again three categories, which is subclinical, acute, and chronic. For the subclinical disease, it's a condition where the disease is present in the organ of the body, but it can't be detected by any abnormal signs or change in the dog. Well, for the acute, or also known as sudden, which the most frequently that occurring in every young dog, is associated with conditions that cause the death of the liver tissue, which subsequently lead to the death of the dog. And the third one is the chronic progressive disease, in which the symptoms are often observed in middle-aged and older dogs with chronic hepatitis and with the damage and the scarring of the liver. Where for the secondary copper hepatopathies, we saw the progressive signs of liver disease due to chronic hepatitis or progressive sclerosis. Liver disease in which the flow of bile is slow or stopped is known as cholestatic liver disease. When both of these primary and secondary hepatotoxicosis displays the symptoms in acute and chronic forms. As for the acute, some of the symptoms are like lethargy, anorexia, depression, and the dog may also show vomiting. And they also mention the yellowish discoloration of the skin and the moist tissue, or also known as jaundice. And also, the dog urine will be dark in color due to the presence of the bilirubin.
What?
Bilirubin.
Really dark in color the urine.
Yeah,
Where will you say biochemistry?
Forgot already.
They also will be like presence of hemoglobin in the urine. Okay. As for the chronic sign, it's more likely the same.
Hemoglobin in the urine?
Yeah,
Oh.
it's very like
That when it is bleeding,
Yeah.
blood
Yeah.
blood urine.
yeah, it's more likely.
So the blood comes from
Yeah.
liver
For the damage in the urine.
Oh, I see.
As for the chronic signs, it's like lethargy, depression, anorexia, and also weight loss due to vomiting and diarrhea of condition. And the dog also may show excessive thirst and urination. And they may also abnormal distension due to the build up in the abdomen and also jaundice as well. Well, for this chronic sign, the dog may also show spontaneous breathing, which may observe in the stool. The stool may be like black in color. Now, for the last sign is the hepatic and subhepatic, which is the nervous system dysfunction due to the liver being unable to break down the ammonia in the body. So that's all.
Um, is there any treatment for this disease? Because it's a genetic disease, but is there any way to suppress the symptoms or suppress the less
Yeah, I'm
reduce
going to
the
be talking about that. And based on what I found, for me, it's not really, the treatment doesn't really cure the disease 100%. It only somehow decrease the symptoms.
Because of
Because
genetic.
it's
Just
quite,
to make it better.
it's quite severe. The disease itself is quite severe.
It has no cure.
No, I wouldn't say it's no cure, but
it can be
it can come back. It can come back. It might be gone for quite some time, but it can come back.
Is it very common among the dogs?
Yeah, like what I said, it's common in the in social also.
Yeah, because
Yeah.
it's inherited and their genetic makeup makes them like easily from the mutation and so on. So they're more likely to get this.
Yeah.
And concerning the prevention, she will explain about it, so you get it. Go
Okay,
there,
so-
yeah, how
Okay,
to
so-
prevent it.
Yeah, the first thing I would talk about is the diagnosis of the disease itself and how we want to find out about the disease. So first, it requires a liver sample of the dog. So they use in the pathology lab where they do research on. So they use atomic absorption to determine the concentration of copper in the liver. So that's how they would determine whether it's healthy or not for the dog. And like what Amir and Pashan said, it is mostly common in badminton breeds, but it may vary as well in certain breeds. Sometimes it's not just because of a genetic disease, it can also be caused by the environment itself. It could be affected by environment. If the dog is exposed to a lot of chemicals, yeah, they can be sort of susceptible to the disease. Okay, so here are the treatments. I found 4 treatments. First is a copper restricted diet. Obviously, everything starts from our diet, right? And then chelating agent, SAME, and lastly, monitoring and maintenance therapy. It's actually a treatment to monitor and maintain the dog. Okay, so first, everything circulates around our diet. So the owner and the vet has to recommend healthy diets for the dog because based on what I read, most the food that dogs mostly consume consists of a little bit of metal, which is shocking for me. Yeah.
That's
Because they need it in their body, but not that much. So certain dog foods, if you do look for the high quality ones, yeah, it's actually really high in all sorts of metal, the heavy ones like zinc and all that. So it's really unhealthy. So you really have to be a responsible owner and buy high quality food for dogs. That's why it's a lot of money to have pets.
Look at
But
the content
it's
before you buy it.
yeah, look at the content and yeah, that's the perks of being a vet. You will learn that.
even for cats.
Yeah.
Especially which have high content in sodium because
Yeah.
kidney disease is really bad.
And the cost
And
to
the
treat
cost.
it, that's the
Yeah.
problem actually. Okay, after restricting the diet, vets usually recommend chelating agents where chelating is when they it's a method of diluting the metal which is copper in the liver. It's a very complex method to treat the disease. So it is very important in decreasing copper accumulation and copper chelators actually reduce liver copper content through like I said, they dilute it and it's a very, very slow process and each time you consult the vet, it can actually cost a lot. Yeah, that's the thing. So the third one is the SAME. The longer term word is S-adenosylmethionine now, which I will just use SAME. It's a precursor of the antioxidant glutathione and it's used as antioxidant in the liver disease and it is recommended to help combat the oxidative effects of copper in copper-associated hepatopathy. Vitamin E is a potent antioxidant commonly used in treating liver disease, particularly copper-associated hepatopathy. So in this method, they use a lot of vitamin E to somehow reduce the copper content in the liver. And the last one is monitoring and maintenance therapy, where this is a really simple thing to do. After all that, after the diet, after chelating agents, and after the intake of vitamin E, both the vet and the owner has to, look over of what the and how the symptoms, is it still obvious symptoms and all that things that when you see normal behavior, then your dog might be in healthy state. If the symptoms come back, then better consult your vet. Okay. So the prevention for this disease is that when one is considering to purchase a badminton breed, which is susceptible to the disease, he or she should ask if the dog's parents have been tested for the gene that causes the type of liver disease. There is also a liver registry available that provides information as to a breeding Badminton's genetic status. Purchasing that Linton puppy from a breeder whose dogs are all free from the problematic genes and markers will diminish the probability of the dog having the disease itself. So I would like to ask if any of you know, are Badminton breeds common in Malaysia? Is it common? What do you think? What are
Not really sure about it.
Malaysia? Okay. No? Have you ever seen it? Has anyone ever seen it in Malaysia? Oh, I don't know how it looks like.
How does it look like? Yeah.
Because I really seen like really properly treated dogs.
Yeah, we don't
because,
really see.
uh,
I feel like we don't really.
in my experience, I often seen strays and that strays that have been taken care by humans. But the most I see is like the small one with long hair which covers the
Oh
face.
yeah, that one.
That one and the Rottweiler.
Oh yeah, police.
Yeah, and the police dog.
Yeah.
That's it. I don't
Yeah,
have
for me
anything.
personally, I think.
I
Oh, that's what it looks like. It's very skinny.
You know that it's dragging his leg.
But for me in Malaysia, people don't really care
care
about the
about
principle to
Yeah.
keep
I
the
think
pet.
it's more to urban countries.
Yeah,
Oh,
but
yeah.
because of the fur, I don't think they will survive. It's really hot here. Yeah.
So,
Yeah, that's really.
so as she said, if you want to buy Benignan terror, better make a good follow-up with the
Yeah,
breeder,
with the breeder.
ensure that the dog is in good condition, the parents, the grandparents do not show any sign of the disease, and then you can buy. So I think that's for all for our group. Thank you all.
Thank you.
Thank you.
Bye. Bye.